This is a production of Cornell University.
Hi, everyone. My name is Chinedozi, and I'm presenting on genetic improvement and chemical characterization of garri quality, of cassava garri quality. So here is my outline. So as Leah said, cassava feeds 100 millions of people. Yeah, so most of these people are in the tropics and subtropical areas of the world. And it originated from South America, and it's being grown like over so many places because of its numerous uses. So cassava is used for food by man. Animal also consume the leaves and is used for industrial raw material, especially in the production of starch and bioethanol. It's a resilient plant. It can grow on marginal soils, usually soils where you can't grow other crops. Cassava can thrive very well in such areas. So it's fastly becoming a crop for food security. And it's also grown mainly for its roots. Because of the high moisture content and cyanide content of cassava, it is usually converted to other forms, so it can have longer shelf life, and the cyanide content can be reduced. So most times it's consumed as chips and boiled, or it's used in making fufu or garri. So fufu is a rated and boiled and pounded cassava, and my work focuses on garri and its derived product called eba. So to make garri, you have to peel the cassava and then wash. After washing, you grind the cassava, dewater and ferment. So this has two variants. Some people dewater and bag the cassava, allow to ferment before dewatering. So in the region where this experiment is taking place, we usually dewater and ferment at the same time. So dewatering is done with a hydraulic press. So it can take like 2 to 3 days, depending on the method and choice. So after dewatering, you filter or sieve into fine particles before roasting with a cast iron pot. And then you have your final product, which is called garri. So then I mentioned eba. So eba is a reconstituted garri. So most people eat eba as meal because garri can be eaten in different forms. Like you can snack on garri and you can actually eat it as a full meal. So to make eba, you have to use a hot water or a boiling water, depends on the kind of garri. So some people before you buy garri, you have to ask the seller if it's hot water kind of garri or boiling water kind of garri. So you have to do that. If not, you get something you never wanted to get. And so why am I interested in garri? So about 70% of cassava is consumed as garri. Garri has a longer shelf life and it's easy to transport compared to other products, and it can be eaten in different forms, as I've said earlier. So cassava breeders over the world have developed disease resistance, high yielding resistance, and varieties that are rich in micronutrient content, but there's a decline in the adoption of these varieties. So one of the hypotheses is that breeders did not incorporate the attributes, especially the sensory and the processing attributes that the consumers want. So most times the consumers and the farmers end up rejecting this or not adopting these varieties. So the objective of my research is to determine the sensory attributes of the two most of garri and its reconstituted products, that is the eba, to develop high phenotyping evaluation alternative for important chemical constituents and the sensory attributes, and to estimate the genotype by environment, interaction, and heritability of garri quality. I also want to determine the relationship between on-farm and on-station measure in a Tricot experiment. So the research is divided into 3 components: the breeding quality study, the sensory evaluation, and the Tricot Anchoring experiment. So for the breeding quality study, is going on right now in 2 locations. The population is the Umudika C2 population, and there are 204 genotypes. I have 2 locations, Umudika and Otobi. There are 2 reps and is an aphalaptis design. So right now data has been collected on the agronomic, disease, and the yield data, yield. And then some genotypes were selected for garri, and they were processed into garri. So for the physicochemical property, I've already, I'm only able to do the dry matter content by a gravimetric and thermometric method. So right now I'm yet to do the other physicochemical properties and then the functional properties. On the screen are the list of the properties I am interested in. Then for the sensory evaluation, I'll be using the flash profiling approach. So flash profiling, as the name implies, is rapid sensory evaluation. So it requires a minimal training of the panelists compared to other sensory evaluation where you have to do rigorous training of the panelists. But the thing is, you need to get panelists that are regular consumers of the product so they can know what to look out for. And these panelists have to be able to explain the senses they get from testing each product. And for flash profiling, you combine, the panelists have to come up with words, so you don't have to give them the word before the evaluation. They come up with words to describe the samples, and then they comparatively evaluate and rank the samples. So it's quite fast. You can evaluate about up to 40 samples. So for me, I'll be, for this experiment, I'll evaluate 40 samples using 20 panelists. Each panelist will be given 5 samples per session, and the sections will be like 2 sections per day. So they'll evaluate like 10 samples per day. So this is just like the schematics for how to evaluate flash profiling. So they generate lexicon. So since different panelists will come up with different terms to describe the samples, after that, they'll gather together, we'll compile each the lexicons and sort out redundancy, and finally they will evaluate the samples, compare the samples, and do the ranking. So this is a sample of the ballot to use for the flash profiling. So they will come up with a list of the attributes, and then they just rate, this is sample A, sample B to the end, and then they rank the samples from low to high. So the next experiment is a Tricot Anchoring experiment. So Tricot is a triadic comparison of technology. So 3 samples are given to farmers to grow on their field. So right now in Nigeria, we have about 300 farmers evaluating 30 genotypes across 4 locations. So this makes, so at the end, they will rank varieties across based on from worst to best, and then they will know which variety is best in each location. And so my experiment is just to anchor that Tricot experiment by just looking at the what the farmer is doing compared to what is going on trial. So I'm going to get like 21 farmers to evaluate the same cassava on the Tricot experiment, and they will take data on different time points and rank the agronomic yield and the garri quality from best to worst, and then they do the rating on a scale of 1 to 5. They'll, like at the end of the whole section, they will rate each product on each variety on a scale of 1 to 5. So right now I just have some of the agronomic data. I'm still working on my data, so I just did a correlation study for each of the location. This is for the garri traits. So I did garri, like the proportion of garri per fresh weight is called garri here. So it's positively correlated with the DMC, but is not, the relationship is not strong. And garri per plot is garri compared to the net plot, and for some reason is negatively correlated with the oven dry method of specific of dry matter content, and is positively correlated with the DMC of using the specific gravity method. And the same thing is what is obtained in Umudika. So I look at the distribution of most of the garri traits, and they are like, they vary across location. The traits vary across location. And so I just picked up two traits to show how they vary across the locations. And then I look at the heritability of some of the agronomic traits and in different locations, and then a combined analysis. So I got for the number of fruits, there's a heritability of 0.48, and a dry matter by oven dry method in both locations was low compared to dry matter by specific gravity method. And then even the combined analysis gave the same low dry matter compared to the specific gravity method. So in summary, the traits vary across location. The heritability for the traits, the heritability also varies with trait and location. And then there's positive correlation, although it's weak, between DMC oven dry method and DMC by specific gravity method. So work is still going on. I have the nearest data, so I'm going to analyze them, and then the Tricot will be due by the end of this month. And then the sensory evaluation, we're yet to do that. So I have to do it when I get back to Nigeria. So I want to acknowledge the NRCRI team for their efforts. This work wouldn't have gone anywhere without them this year. I'm so grateful. And I also want to thank my supervisory committee and Next Gen Cassava and every other person. Thank you. So I welcome questions.
Thanks for the wonderful talk, Chinedozi.
Yeah,
so you can either write on your questions in the chat, or you can also raise your hand and I will ask you to unmute, or just unmute if you want to do that.
Hey, Chinedozi. I had a question about the fermentation step.
Okay.
So what microbes contribute to that fermentation, and is there a genetic variance to that? Like, do different varieties have different inoculums, or is that an inoculum source that's added to the production line? How does that work?
So normally the fermentation takes place naturally. You don't add anything. So, but for my research is not I'm not like, this is far beyond the scope of this work. We're not looking at the inoculum and the bacteria or the fermentation process. But before you get garri, it has to pass through fermentation process. Yeah, but it's natural fermentation. You don't have to add bacteria or some stuff.
Okay, I'm just curious.
Yeah, it's natural. All right, thank you.
Hi, Chinedozi. I have a quick question. Thanks for your interesting seminar. You mentioned there's hot water versus boiling water,
Yes.
EBA. Is that a genetic varietal difference? Is there nutrient composition, or is it something with the processing?
So we are yet to know that. We don't know what causes it. So I think that's part of this research. I hope at the end of the research, we'll know why you need to have different temperature water to make different kind of EBA or different kind of garri.
Interesting, thanks.
Sorry, this is Jean-Luc. Interesting question. Did you mean, do people do the one kind, do hot water as opposed to boiling water with some varieties and boiling water with other varieties, or what? I'm not sure I understood your question.
Right, so I think what I'd heard was that when you went to the market, you were told what you were buying was either to be boiling or hot water. Maybe I misinterpreted. And so I was wondering what caused, the person at the market selling it to determine something was one or the other.
Yeah, so most times these people that sell must have used it, because some sellers buy while others process themselves and sell. So they already know, because they've already tried it in their houses, and they know that this is not good for hot water, or this is good for boiling water. So they have to tell you. If you don't ask them, they just give you, and then when you go home and try it, you realize yourself that it is for hot water and it's otherwise. Yeah, so right now we don't know what causes that characteristics in garri. So we are still working on that.
Great. Thank you so much. That was a wonderful talk, and the last talk of our seminar series for the fall semester.
This has been a production of Cornell University, on the web at cornell.edu.